Okay. Good morning, everybody. Okay. Okay, so we're going to talk about vision this week. But before I get started, we were talking about measurement last week. And we were talking about spectrophotometers and colorimeters, right? Everyone remember what's the difference between a spectrophotometer? What does a colorimeter do? Yeah. Right. So it tries to mimic the visual response to that particular spectral distribution, right? As opposed to a spectrophotometer, which gives you the energy or the relative energy at different wavelengths. So, RIT, spares no expense to keep your education state of the art, right? And so we just recently acquired a really advanced colorimeter, and I wanted to demonstrate it for you today. And so it's right here. It's got a speech synthesis function even in it. And so here's how it works. It's really advanced. You point the arrow toward the color you want to measure. You pull this arm down. See, the juice is orange. Purple. Incredible, huh? Amazing. Okay. That was a garage sale item this weekend. So, okay. So with that, there was a request to go over one of the calculations that you're supposed to be doing for the project, okay? Relating to measuring the mean color difference from the mean and the precision for the measurements. So I'm happy to do that. So, well, I guess maybe I can start here. Well, maybe I'll start with Burns, right? So here's the table from Burns that you guys are going to be replicating in some sense. And the purpose here is to figure out, okay, you're making a set of repeated measurements of differences between a standard and a sample, okay? And you want to know how many repeated measurements do you need to make in order to get a certain level of precision in your measurement of the difference, okay? And so the data going into this whole process is a set of measurements, and the measurements of the difference between the standard and the sample, okay? So you see here there are 10 measurements right here, right? 10 different measurements. And the measures are delta L, right? The difference in lightness, the difference in chroma, and the difference in hue angle, okay? So these are all difference measurements. So in your own calculations, you're going to need to measure a standard and measure a sample. Yes?
I know there was a way to check whether or not H was considered negative or positive.
Yes.
Is there a similar thing for L, or is that just the difference?
No, L is the only one I think I can go. We'll go through the document, and it says the only one that has a sign associated with it, I think, is the H.
So
Okay.
It's like the absolute value?
Let me see. Let me just go through, and we'll get to it. Okay. So, so these are all difference measurements between a standard and a sample. Okay. So what are you supposed to do? You're supposed to measure.
Where's the first convert from LEV to the H and C?
Yes, you have to calculate those, but I forget. So do you actually have a standard that you can measure, or do you have to just measure? Okay. So then.
Just like the average of all of our values. I can call that the same.
Did you make it? You did 20 measurements? You did 10. Okay, so what I did in the spreadsheet was I actually had a standard and a sample, but what you could do is you could just do 20 measurements on your sample and call one of them the standard and one of them the.
Oh, so you don't have to do the average.
No, you have to take the average, but you want 10 differences going in. okay, not 5 differences going in. If you did 10 measurements, you'd only have 5 differences, right? Do you understand?
Yeah, I was doing it different. I took 10 samples and then I averaged them all, and then I took the difference from that average to each sample. So I had 10 from the median.
You're measuring different samples? Or,
They're all, yeah, I took, like, we used the red one.
okay,
The red one,
yes,
the blue one.
okay,
So I had 10 measurements of the red.
yes,
And then I averaged all of those.
yes.
And called that my sample, because it was like the average of every single measurement.
Okay, yeah.
And then I measured the difference from
Oh,
each
yeah,
sample
okay,
to.
that's fine.
Is that okay?
That's fine, yep. Okay. Okay, so you have 10 difference measurements going in, right? So now, from those difference measurements, you calculate the average, right, of the differences, and you calculate the standard deviation of those differences. And those come over here, okay, so the standard deviation in particular. Then you define kind of arbitrarily, based on your application, a tolerance, upper tolerance and a lower tolerance, right, on the precision, and a tolerance range. So the upper tolerance is +2, let's say, and -2, and so the range is 4, okay? And for the purposes of this exercise, I just said leave these the same as in Burns, right? So give yourself a +2 and a -2 here for a tolerance range of 4, a +1, -1 in delta C for a range of 2. Come back. I guess I have to plug in. I know, I just have to plug in so it doesn't keep doing that. Bear with me 1 second. Okay. All right, so now you have your tolerance range, right? And then, you know, a rule of thumb from industry is that you want your the goal for your for the precision in your measurement is going to be 10%, right, of your tolerance range. So here you multiply the tolerance range times 0.1, and you get 0.4, 0.2, and 0.1. But then also the industry says, okay, well, 0.1 might be too small, so let's just have the bottom of the tolerance range be 0.2, okay? And so this one stays as 0.4, this one stays as 0.2, and this one gets rounded up to 0.2, okay? So all this in here, well, this is set by your application, right? And this is kind of set by convention, right, in industry. Okay, so now you have these numbers going into the process. You want to calculate what the sampling number is, right? And the sampling number is given by the ratio, right, squared ratio of the standard deviation in your measurements over the sampling, the standard error, right? So now where do we get standard error from? Standard error comes from here, okay? So this is the goal for your standard error. So you have, in this case, you have 0.45, right, over 0.4 squared is going to give you the number that you need to, the number of samples that you need to do to achieve a certain precision, the standard error, okay? And so when you do that calculation, this n comes out to be 1.27 for delta L, 3 for delta C, and 0.5 for delta H. And then because you can't make partial fractional measurements, you round up, okay? So this rounds up to 2, this rounds up to 4, and this rounds up to 1, okay? And then because you presumably would make one measurement of all the values at once, you take the biggest one, and so that says, okay, if you want to achieve this level of precision, right, in your measurements, then you need to make 4 measurements, 4 repeated measurements on your sample. Okay? Yes?
Why do you round up to 3.06?
Because you can't make .06 measurements on a sample, right?
Then how do you get from rounding up to 1 and then requiring 4 samples?
Oh, from here to here? So, okay, so this is just the pure calculation, right? And it says to achieve this level of precision, this one, you need to do 1.27 measurements on that sample. Okay, well you can't do 1.27 measurements, so you have to do two measurements. Okay, same here. 3.06 gets rounded up to 4, and 0.56 gets rounded up to 1. Okay, now you could, if you were measuring each of these things separately, you could do 2 measurements to get this precision, 4 measurements to get this precision, and 1 measurement to get this precision. Okay, but in practice, you really have one instrument. You're getting all the data at once, so it's the worst case. You have to use the worst case. Okay, if somehow you were doing some other kind of instrument or something that measured these things separately, you could use this line, but typically you'll use that line. Okay? All right. So that's the process. And let's see. And I made this spreadsheet to help you out without the formulas, unfortunately. And I realized, as I went back and looked at this, that when I printed out the spreadsheet to put it in this slide, it was cut off from here. So I put it back in, and I updated it in my courses. But so this is the sampling number calculations are over here. Okay, this is the mean color difference from the mean calculations over here. Okay, so now what the table that you see here is the same as the table that I just showed you in Burns. The only difference, and the reason there's space in here, is because the data going into Burns is already deltas. Okay, it's already difference measurements, and I didn't have those difference measurements, so I actually had to measure a standard and a sample. Okay, and so just in the spreadsheet here, the odd rows are the standard measurement, and the even rows are the sample measurement. Okay, and so then when you do the differences between them, you get these spaces in the rows. Okay, so each one of these, so this row here represents the delta or the difference between the first standard and the first sample. Okay, this is the difference between the second standard and the second sample, and so on. Okay. Okay. Oh no, sorry, that's not. This is the first one here. This is the second one here. And this is the mean of all the standards. Okay, so I did what you did. I took the mean of all the standards, and I used that as my mean to figure out the differences with respect to each sample. Okay? So is there anything about this that's not clear? Yes?
The only thing I'm struggling with is getting from A, B to C.
Okay. Yep. All right. So in the resources, I put a document, right? This one here. And it talks all about LCH, right? And so blah, 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 blah. Goes down. Where's the equation I need? Okay, so the first equation you need is C star, which is just the square root of A star squared plus B star squared. Okay? So that's your C star.
Okay, yeah, I agree with that. I was just wondering
Okay.
if there were more down that were like X's and Y's, and I do those. I only use
Yeah.
A's and B's.
Nope. As long as you have A's and B's, you can use those A's and B's. You don't have to go. I mean, this usually what you're doing is you start from XYZs. You calculate you use the XYZs to calculate the A's and B's, and then you calculate these from the A's and B's. And we'll talk all about that next week.
So, because I know because I got the measurement for XYZ.
Yeah.
So I didn't know if I had to use that.
No. You can use the LABs. Yeah.
Actually,
Right. Okay. And so then the other bit where I had to actually read the fine print was there's this this intermediate calculation you have to do where you get small h, right, here, which is the arctan of b star over a star. And then to calculate the big H, the difference in hue angle, you use this equation here, okay? So you have delta H, big H, is equal to the square root of delta E squared minus delta L squared minus delta C squared. And then there's this fine print which says if the hue angle of the sample is greater than the hue angle of the standard, then delta H is positive. If it's less, then delta H is negative, okay? So in your calculations, you want to make sure that you somehow adjust the sign of delta H so that it comes out according to these rules, okay? And if you go back here, let's see, if you go back here, you'll notice delta H, right, is sometimes negative. Actually, it's, yeah, 2 cases it's negative, but otherwise it's positive. And there's a sign, I don't know if you're using Excel, but there's a sign function in Excel where you can take the difference between 2 things, and if the sign comes out positive, you can assign the sign to the result. So anyway, that's just something to keep track of as you're doing this. Okay? Other questions? Everyone good? Yes?
So since we don't have a standard.
Yes.
20 measurements and I just use 10 of them as the standard.
Right. That's right. So just like, you know, move the thing around like this on the sample and just pretend, right? Okay. Okay. Everybody had got Argyle installed and got the monkeys going and stuff? Yes?
What is the MCDF calculations and the breakpoint column?
That's this one. So you're supposed to do 2 calculations on the on the data. One is the mean color difference from the mean, which is how much each individual measurement varies from the mean of the measurements. Okay, and you can just calculate it. You take the average of all the measurements, right? That's the A bars, right? And then you calculate the difference, the squared difference of each individual measurement from the average, sum them up and take the average of that. Okay, so, and it's all in the chapter. Okay. All right, well, I can see I'm not going to get through the lecture today, but that's good. It's fine. All right. Okay. Right. So this week we're talking about color vision, and presumably this is a subject that you are, well, if not color vision, at least you're somewhat familiar with vision, right? So let me just ask. Anyone know what the macula is? Yeah.
Isn't that the yellow filter?
Yes. Okay. Good. What's a receptive field?
Just the diagram?
It's a diagram. Okay. Now what's a yeah? Okay. Sorry. That's a good start. What's a receptive field?
Due to processing in the ganglion cells.
Right.
And it functions as a sort of edge detection.
Okay. 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 And contrast sensitivity function? What's that? Yeah.
It's like the FDF for
Okay. All right. All right. What do you know about adaptation? Yeah.
There's darkness, lightness, and chromatic adaptation.
Okay. Good. Good. What are the things that contribute to your adaptation? Like, what are the mechanisms of adaptation? Okay, so rods and cones, one. Anything else? Pupil, okay. Sorry?
Would be your iris then.
Yeah, so iris and pupil, yep. That's true, but that's not like a mechanism, right? What's the thing that's underlying that in the visual system? Ion channels. Okay, yeah, so the bleaching and the regeneration of the photopigments, right? And then what's the last one? So we have rods and cones, right? We have pupil. We have photopigments, bleaching and regeneration. And then probably the last and the most important one is just the neural network itself, right? That the whole system is designed to have this kind of automatic gain control. Okay, good. Good. Jeff has taught you well. All right. Okay, so I'm starting off with some illusions just because I can, because you know that's the whole point of studying vision, is that you can put up these cool illusions and people go, wow. Okay, so have you seen this one before? Okay. No motion there. There's no motion. And you've seen this one before, of course. A and B, right? White, light square, dark square. No, in fact, they're the same color. Okay. This one I like. Let's see. Have you seen this one before? Okay. All right, so first the ball rolls along the ground, then the ball goes up in the air, right? No, Ball's always doing the same thing. The ball's always going the same place. See that? Okay. The reason you see it as moving differently is because of its relationship to the shadow. Okay? All right. And then this one I like. So here's a teapot, right? Shiny teapot rotating. And now you stick the reflection to the teapot and it looks like it's painted, right? Now you make it move and it looks shiny, and you stick it to the surface and it looks like it's painted, okay? And the other cool thing about this is that now that the teapot, this is another one of those things where the teapot is actually always moving exactly the same way. I don't know how to point to it here, but the rotation of the object is always the same, but you perceive it to be rotating differently when it's in one state or another. So here, it's like rotating around the vertical axis, right? And in the other case, it's rotating about the horizontal axis. Okay, so there's interesting correlations there between motion and material, and and then have you seen this one? Okay, you all remember George Bush, right? That winning smile. Okay, so anyway, okay, now enough. All right, so okay, so this is what we're going to talk about this week. Basic outline of vision. Some of it you will have, you'll be familiar with. Some of it you may not be. If there are questions, let me know. So. All right, so there's the eye. You all know the eye. So in the front of the eye is the optics, right? The optical system of the eye, the cornea, the iris, and the lens. The cornea is fixed, provides most of the refractive power of the visual system. The lens is not fixed and changes shape to, provide accommodation and focus at different distances. And the iris opens and closes to allow more light in. So in the back of the retina is a bunch of stuff. The outer coating of the eyeball is the sclera. It's kind of like a tennis ball. If you've ever cut open a tennis ball, you know how it's all fibrous and like that. So it's tough and keeps the eyeball in shape. Inside that outer coating is the choroid, which is where there's a blood supply to the retina, right? So the retina is very energy intensive. It needs a constant supply of blood and nutrients to keep going. And the retina is where, of course, all the photoreceptors are. In the center, near center of the retina, near the optic axis is the fovea. and the fovea has the highest density of photoreceptors, right? Cones in particular. And then some distance off the optic axis, about 20 degrees, is the optic nerve, where all the fibers from the ganglion cells, which we'll talk about in a minute, converge and exit the eyeball. And there are no photoreceptors in the near the optic nerve, which means it's called the blind spot. Very good. Okay, so we talked about the macula. I asked you about the macula. So in the center of the visual field, right, there's the fovea. There's a diagram of the fovea and cross-section. And the fovea has the highest density of rods and cones, right? Well, it's actually the highest density of cones. There's no rods in the actual center of the fovea. But highest density of photoreceptors and consequently the highest acuity vision. How big is your fovea? Two degrees, right? Which is why there are 2-degree standards, right? 2-degree standard in color measurement. Okay, so the interesting thing about the fovea, and actually we'll talk about this later, is that it's a place where there's highest density of photoreceptors. It's also a place where all the other stuff, all the other neurons are pulled aside, so the photoreceptors are most exposed. And that's good and bad. It's good in the sense that it allows most of the light to get through without diffusion. The bad thing is that it allows most of the light to get through without diffusion. And so one of the things about light is both good and bad, right? Light is what we need to see, but light can also damage. It damages everything, right? It's sunlight. You know, you put something out in the sunlight and it fades over time. Well, the same thing is happening with light in your eye. It's there's an issue called phototoxicity, which is the study of the idea that light is damaging to various structures, right? I mean, you look at your skin, right? You get freckles and so on your skin, and the same thing is happening to your eyes. And so over the eons, whatever, the eye has developed a protective system, the macula, right? And so covering the fovea and a little beyond is the macular pigment, right? The macular pigment is blue absorbing, absorbs short wavelengths, okay? And the short wavelengths are the most energetic, and they're the ones that do the most damage to the neural structures, okay? So, you know, I took all this off the web, so take it with a grain of salt. But, you know, you can think of the macula as internal sunglasses, right? So here's the various wavelengths of light. The light that is most damaging to your eyes, or potentially damaging, is short wavelength visible, right? And this is called the blue light hazard. And the macula absorbs just in this region, okay? Absorbs most just in this region, so it protects your fovea from excessive blue light. Well, it just so happens that there are two, you actually can't see it here, but there are two chemicals, right, in the macula, lutein and blah, blah. I don't know how to pronounce it. It starts with an X. Yeah, xanthanthin, lutein and xanthanthin. And it just so happens that one of the things in the world that supplies lutein and xanthanthin is kale. So you want to protect your eyes, eat more kale, okay? So anyway, it just shows here that, you know, these are these various supplements, and those are the absorptions of those supplements, and they can protect your eyes from the degeneration due to blue light. Okay, the thing that's interesting with respect to color vision is that, and Mark talks about this in his chapter, is that differences in the macular pigment are the biggest factor in individual variations in color perception. Okay, so some people have very thick macular pigments, others have thin, different densities. Okay. Okay, so then there's the photoreceptors themselves. In each eye, there's about 130 million photoreceptors, 120 million rods, and about 7 or 8 million cones. I probably don't have to do this, right? You know, the cones are for daylight or photopic vision, right? The rods are for nighttime scotopic vision, right? And they're called rods and cones because of the different shapes of their outer segments there. Okay. The thing that's important for us studying color vision is that the photoreceptors have different spectral sensitivities. Okay, and so here are this is a diagram of the distribution of rods, or distribution of cones in the fovea, and you can see that there's kind of a random arrangement. There are much more long and middle wavelength, red and blue, red and green, than there are short wavelength blue cones, and they have this kind of pseudo-random arrangement. So here you can see the spectral sensitivity curves of the different cone types and also the rods. The rods have there's a single kind of rod, right? And it has a spectral sensitivity curve that looks very much like the cones, but it peaks at a different location, peaks at about 498, 500 nanometers, okay? There are three types of cones that have different spectral sensitivity curves. And here you can see that they peak in different locations across the spectrum, right? So there's a big difference between the middle wavelength and the long wavelength cones and the short wavelength. So the short wavelength system and the middle long wavelength system are distinct from one another, and they're distinct evolutionarily, right? So people who study the development of the visual system think that initially we were dichromatic, okay? So there was a short wavelength system and a longer wavelength system. And then over time, that longer wavelength system split into two, okay? And so that's why the peaks of the middle and the long wavelength system are so close to one another, and that the short is distinct from it, okay? And people who speculate even further go to say that, okay, well, the short wavelength system, if you talk about differences between these two, you're talking about kind of a yellow versus blue, right? So sunlight versus skylight kind of distinction. And if you talk about the difference between these two, you're talking about red-green. So you're talking about ripe fruit versus, green fruit, or leaves versus fruit, that kind of thing, okay? Okay, so but the significant thing about this is that there are three kinds of cones, right? They're sensitive to different ranges, and they have broadband overlapping spectral response properties. Okay, so again, a reminder, right? So there's the receptors are not equally distributed across the retina. Okay, in the center of the retina, there's the fovea, right? And the fovea has the highest density of cones. You can see this curve here, right? So most of the cones are the highest density of cones is in the fovea, and then the density of cones drops off very quickly as you get out to the periphery. And by 10 degrees in either direction, it's pretty much at its base level. Conversely, there are no rods in the central fovea, right? And the density of rods increases as you go out in the periphery and peaks at about 20 degrees out in the periphery, okay? So, the story everyone always says is, well, how do you know there's no rods in the fovea? Well, if you're outside looking at stars at night, right, you don't look at the star. If you look at the star, you can't see it. You should look off to the side. And the best way, is about 20 degrees off. And then you'll have the highest sensitivity, okay, because of your rod vision. And so that's the diagram of the distribution of rods and cones. this is what the different pieces of the retina look like. So in the fovea, very high density of cones. As you go out toward the periphery, you get rods. You get, so the density here is these are these are cones. The cones are the things in the center area surrounded by lots and lots and lots of rods. Okay? So there's 10 times, 12 times more rods than there are cones. So, all right. So this is interesting. Several years ago now, I think 10 or 15 years ago, David Williams, who's at the University of Rochester, developed some systems of adaptive optics to allow imaging, direct imaging of the retina, okay, of the photoreceptors in the retina. And so these are some pictures that he took. They're color-coded, but they're pictures of real people's retinas. And what he found was that there's big differences in the distributions of rods, so distributions of cones in different individuals' retinas, okay? And so these are all 4 different people with normal color vision, okay? But you can see that there's huge differences in the ratio of the long to medium wavelength cones, right? So the red versus the green. So this person has a ratio of 1.1 to 2.7. This person has a ratio of 16.5 to 1, okay? So mostly green, not a lot of red, mostly red, not a lot of green, but still they have normal color vision, okay? So there's a lot of processing going on after the photoreceptors in order to give you your experience of color that can compensate for these huge differences in the front end, right, in the sensor. Okay. The thing that is pretty constant across everyone, though, is the ratio of short wavelength cones to middle and long. So everyone in this picture has about the same number of short wavelength cones. Okay, so that's not varying. Okay, so once we get past the photoreceptors, then we have this neural network in the retina that are called the plexiform layers, right? Plexiform layers. And so here are the photoreceptors, and as you all know, the curiosity is that the retina is organized backwards, right? So the photoreceptors, like here's the back of your eyeball, right? And so the photoreceptors point down like this, and light comes from here and has to go through all the neural junk before it gets to the photoreceptors. And why is the eye? Anyone have any sense? Yes.
Isn't it to keep the photoreceptors nourished?
Yes. Okay. So that one reason is that the photoreceptors require a lot of energy and nutrients, right? And so keeping it close to the choroid, right, which is back here, allows that to function better. Any other thoughts? Well, sometime long ago, we were nocturnal, right? Or our ancestors were nocturnal. And so if you look at animals that are still nocturnal, they have the same organization of receptors pointed toward the back of the eyeball. But instead of having the pigmented epithelium here, which is dark in us and absorbs light for the most part, they have something called the tapetum, right, which is a reflective surface. And so what happens is light comes through, gets absorbed in some part on the first pass, then reflects off the tapetum and gets absorbed on the second pass. Okay, so it's an efficiency, organized for efficiency, right, in collecting light for nocturnal animals. Anyway, it's what we have. So this is the organization, right? So the photoreceptors, the rods and the cones here are pointed back toward the epithelium. And then they are connected to each other by a bunch what are called bipolar cells, right? And the bipolar cells and the receptors are connected by horizontal cells, and these and then further on up the chain, the bipolar cells, the green ones, are connected to the ganglion cells by the amacrine cells over there, sort of diffuse network of cells. And the ganglion cells, the axons of those ganglion cells form the optic nerve, okay? So all this stuff here, all this junk that's connecting the photoreceptors to the ganglion cells is what creates the receptive fields, okay? That are the primary processing units in the retina. Yes?
I don't think it's quite understand how having a reflective surface instead of the pigmented epithelium makes you nocturnal.
It doesn't make you nocturnal. I was just saying that one of the differences between nocturnal animals and us is that in us, the epithelium is dark and actually absorbs light that hits it, okay? For the most part, you know, the red eye that you see is some reflection from that, but for the most part, it's dark, okay? In cats, for example, that they have, instead of the epithelium, they have something called the tapetum, which is essentially like a mirror, okay? So when light comes through, sometimes the light gets absorbed by the receptor on the first time past the receptor. If it doesn't, then it has a chance of reflecting off the tapetum and getting absorbed on the second time through, okay? So all I was saying is that nocturnal animals have this mirrored back of the eyeball in order to maximize light capture, right? It doesn't make them nocturnal, it just means that they can get by better in low light conditions. Yeah. Okay. So so there's this neural network, right, in the retina, and as I say here, it's involved in doing several things. One is doing kind of automatic gain control and adaptation, right? Performs image processing functions like you were talking about edge detection, right? So or something like edge detection, multi-frequency decomposition, right? So you get different size receptive fields are coding for different scale features in the image, all this kind of stuff, okay? And we'll talk a little bit about that. Okay. So I'm assuming all this is familiar, right? This is all like review, okay. All right, so there's the ganglion cells. Again, there's 130 million photoreceptors. They converge down to about 1 million retinal ganglion cells, okay? And there's the receptive field of a ganglion cell. This is a ganglion cell down here. And there's the photoreceptors that are connected to the ganglion cells. And the receptive fields have this circularly symmetric organization, right? So there's the center and the surround, right? The annulus. The annulus is the outside part and the center, okay? And in general, the organization of the receptive field is that there's this antagonism. There's this positive-negative relationship between the center and the surround, okay? So that stimulation in the center may cause a positive response from the ganglion cell. Stimulation in the surround will cause a negative response, and then stimulation of the whole thing will neutralize the response, okay? So that's a basic organizational property of the visual system, okay? So here's again, this ganglion cell, right, sort of diagram. This is the functional diagram of this ganglion cell receptive field. And there are basically two kinds of organizations, right? They're both antagonistic, so the center and the surround are polar opposites of one another. The one where you get a positive response from stimulation in the center is called the on center, right? And if you get a negative response with stimulation in the center, it's called an off center, okay? So those are on center and off center receptive fields. So those are the two basic flavors. One of the things that to understand is that there are these ganglion cells, or so the receptive fields, all over the retina, okay, but they are not uniform across the retina. So one of the things that happens is as you go out from the center, from the fovea to the periphery, the average size of the receptive field increases. Okay, so very small receptive fields in general in the fovea, right, where there's dense photoreceptors, and then the size of the receptive field increases as you go out toward the periphery. Okay, so that's the general trend, but at every location there are receptive fields of many different sizes. Okay, so in the fovea, even, you'll have very small receptive fields, and then you have larger and larger and larger fields, and the same at every location. And so thinking in terms of image processing, what's happening is you have these receptive fields. The receptive fields have different sizes, which makes them sensitive to features of different spatial sizes. Okay, so if you think about doing an image pyramid, right, or a MIP map, they used to call them. Okay, so taking the input and decomposing it into a series of bands, band-pass images that have different features, or features of different spatial scales in them. Okay, that's kind of what's going on at the first level of coding in the visual system. All right. Okay, so in addition to the frequency decomposition, the receptive fields, many of them are spectrally opponent. So in addition to being on-center, off-center, some of them are yellow-blue or red-green. Okay, in their sensitivity, and they also have varying temporal properties. Right, some respond very quickly, others respond, or some respond to high-frequency changes, others respond best to low-frequency changes. Yeah.
I know that they vary in size.
Yeah.
But is there like kind of like an average amount of photoreceptors, or is it that you said there's 130?
Yeah. So the one anchor that I know is that in the fovea, there are receptive fields that have one cone in the center, and then some number of opponents in the so that's the smallest. So they get as small as one cone. In the periphery, you're dealing mostly with rods way out there, and I know that there can be hundreds of rods in the center of a receptive field. So it can be that much, right? But specifically, I don't know beyond that.
Yeah,
Yeah.
so we could have a lot of receptive fields.
Oh yeah, well I mean, so you should think of the retina as like coated with receptive fields, right? And that it's not that so an individual photoreceptor can be part of many different receptive fields, right? So, yeah. Okay, so once we get out of the eyeball, it actually gets even more interesting. So here's the retinas, right, up there at the top. And then the ganglion cells pull together. The axons pull together and form the optic nerve, okay? And the optic nerves exit the eyeball. And then they travel along until they reach something called the optic chiasm, right? And the optic chiasm is a place in the brain where half of the fibers from the right eye cross over and go to the opposite hemisphere of the brain, and similarly with the left eye, right? And so what you have happening is you have an organization changing where the retina, we will always like to think of the eye as a camera, right? So the retina, you have this retinal surface and the image is coming in, and you have the image being coded by the retina. But now when you get here, what happens is that image gets broken apart, and half of it goes to the left hemisphere and half of it goes to the right hemisphere. So rather than being coded as a unique image, what ends up getting coded in the two hemispheres is the right visual field and the left visual field, okay? So the left hemisphere is actually coding all the information. Well, let's see. I don't even, so yeah, the right, the left hemisphere is coding stuff that's coming in the right visual field, and the right hemisphere is coding stuff that's coming in the left visual field, okay? So if you see people who have strokes, often they'll lose stuff, you know, to the left of them, and it's because of this hemispherical organization, okay? Okay, so first thing happens is that half of the fibers cross over. So we have the right visual field here, the left visual field here, and these fibers converge on the lateral geniculate, okay, which is a sort of a processing station, first level of processing. We'll talk about what the LGN does in a minute. There's the superior colliculus. What does the superior colliculus do? Jeff must tell you, come on. It's his baby. This is his part of the brain.
I don't think he ever mentioned that.
Really? It's eye movements. This is everything to do with eye movements, the superior colliculus. Okay, anyway. And then the fibers from the LGN go on to the visual cortex, okay? So this is the first place where we see the breakdown of, and you'll see it as we go further on, of this eye as camera analogy, right? So the first thing that's happening is that, you know, the retinal image is getting split up into left and right visual fields, and then as we go further on, it gets broken up and broken up even further, okay? All right, so what does the LGN do? Well, okay, so there's the left and right eye, the right visual field. The fibers come to the optic chiasm, and they converge upon the LGN, the lateral geniculate nucleus, in the right hemisphere, okay? So the LGN has this layered structure to it, and the fibers come in and they intermingle like this, okay, in the layers. So there are 6 layers in the LGN. 2 of these layers are called magnocellular, and 4 are called parvocellular. Magnocellular just means big cell. Parvocellular just means small cell. Okay. So have you heard about magno-parvo before? What is it? Tell me about the magno and parvo systems. Anyone? No? Okay. Okay, so this is interesting. Okay, so there are these cells in the LGN, and they are divided into these 2 classes: the magno cells and the parvo cells, right? And the magno cells and the parvo cells are different in a variety of ways. So the magno cells are big, the parvo cells are small. The magno cells are generally come from the periphery of the visual system. The parvo cells tend to be coming from the center of the visual system, okay? The spatial resolution of the magno cells is low, and so they're gathering, the receptive fields are very big, okay? The spatial resolution of the parvo system is very high, so the receptive fields are generally small. The temporal resolution of the magno system is high, so it's very sensitive to changes in light intensity or an edge coming across it, okay? Conversely, the temporal resolution of the parvo system is low, okay? So it's not very sensitive to temporal changes. And the magno system is largely achromatic, and the parvo system is color opponent. Yes?
How's that different, like the magno being like rods and parvo being like?
No, it's not rods and cones.
Very, like, seems like a very similar split of descriptions.
I guess there's some similarity there, but it's not that it's divided up by the type of photoreceptor. Okay, so there are cones going into the magno system, right? There are rods going into the parvo system. So it's much more of a functional distinction, right? So that's why I put 2 visual systems here, right? So the way that it's generally described is that this is the detect the tiger system, okay, right? So you're a monkey hanging out in the jungle, right? And so you want to see if there's a tiger or a jaguar or something coming at you. What do you need? You need something which can detect change, right? It doesn't have to see color, but it has to be very sensitive to change, to some sort of movement, okay? So this system is very good at detecting movement. On the other hand, if you're trying to find those berries in the tree, right, you need high resolution to see detail, and you need color, right, in order to distinguish between the leaves and the berries, right? This also is associated with animals that are more socially attuned, right? So face recognition and that sort of thing is in part of the parvo system. So, okay, so again, here's another place where there's this breakdown of the idea of the visual system as a camera, right? So if you think about the visual system in this way, there are, in a sense, two visual systems, right, superimposed on each other at the same time, right? There's the one that is very sensitive to motion and is detecting, change, and there's the other one that's organized for seeing fine detail and doing color discrimination, okay? And so in terms of evolution, it's interesting that, that this one piece of hardware, right, one piece of sensor has evolved into these two systems, right? Okay, so once we get further on into the cortex, right, so the fibers come from the retina to the optic chiasm to the LGN, the lateral geniculate, and then from the lateral geniculate to the occipital cortex, right, the visual cortex in the back of your head. So this is visual cortex back here, primary visual cortex, right? Primary visual cortex is also known as V1, right? And the people who do, the physiology have named the different regions of cortex that are associated with vision, V1, V2, V3, V4, and so on. But V1 is the primary cortex back here, and it's the first one to get the projections from the eye itself. Okay. So what happens in the cortex? Well, the cortex is this kind of very well-organized structure. It's got layers like this. It's got a whole bunch of different layers, and it's got columns of fibers going down. So it's very, rectilinear. And what happens in the primary visual cortex is that the input from the LGN, from the magno and the parvo system, gets split into these different layers, and there's 8 different layers that people have identified. And these layers are tuned to code for different features in the image, right? And so, and by doing electrophysiological studies, people have figured out that okay, layers 2 and 3 code for color, layer 1 codes for shape, for form, layer 4 B codes for motion, right? And so what's happening is the input from the eye, from the more peripheral stuff, is getting coded into these different maps. Okay. Yep.
If it's going through all these different areas,
Yep.
it sounds like sometimes it's going like one right after the other rather than at the same time. And what happens when one of those gets messed up? Like someone can't perceive like motion or something.
Right.
That mess up your color perception.
So whenever there's some kind of brain damage, right, it's always, I think it's always very complicated, and each individual is very different from the next individual, right? So it's hard to say what's going to happen. okay, so in human beings, people can lose lots of stuff at once, or they can lose very specific stuff. Okay, so and now as I'm thinking out loud here, as I'm trying to answer your question, I know from studies done with monkeys, right? There they can go in and they can cut out certain pieces and see, if it affects just one thing. And so they do experiments where they somehow knock out one layer, and it only knocks out that one function and leaves everything else pretty much the same, right? Now the monkeys are messed up, right? They're not running around, but in terms of the tests that are being done, they find that they still have relatively normal, color function, and that's in fact how they figured out, that these different layers are doing these different things, right? Yeah. Okay, so the high concept here is that, the input is getting split into different maps that are coding different features, right? Motion, color, form, disparity, right? And then as you go from primary visual cortex, you go into higher levels where these primary maps now get related to one another, okay? So you may have a map that takes stereo and motion and puts them together or compares them to each other, or form and color and compares them to each other. And so as you go higher and higher up in the visual cortex, you get more and more of this comparison and reconstruction in some sense of what's out there in the world. But the interesting thing is that, the visual system is taking what's out there, right? And first of all, it's compressing it a lot, right? Because from the retina, you have 130 million receptors. You're doing this edge detection and color compression to get down to 1 million optic nerves, right? Then it comes through the optic nerve, it gets split in the LGN into this achromatic motion sensitive system and a chromatic, shape detail sensitive system. And then those things get broken down into form and color and motion, and disparity. And then those things get related to one another and get built back up into what you see, right? And so the take-home message from that is that, our perception is highly processed, right? It's really a construction that comes after a lot of breakdown into elements and put back together again. And in that, there's the opportunity for other stuff from the brain to come in and sort of fill in and decide how to hook things up together again, decide what to pay attention to, right? And so that's what I said here is the inputs, or those maps, those factors, are recombined at higher cortical levels with executive influences. So, what your experience is, what you remember, what your goals are, what the task is that you're trying to do, right? And then also primitive influences like emotions and drives, right? The lizard brain, right? The thing that makes you see, when you see a red, you go, oh, must mean danger. Red means danger, right? So there's all that going on in your visual system, right? In your perception. So it's very much a construct. So the last thing about the physiology is that from the visual cortex, the processing in the visual cortex also gets split into two main streams that go into higher parts of the cortex. There's the ventral pathway, which goes down the side of your brain here, and that is generally thought to be involved with detection and identification. And connection to memory, right? And it's generally called the what system. So identification is its function. And then there's a dorsal system, right, up here, dorsal, like dorsal fin, and that's the where system. So it's like, where in my environment is this stimulus coming from, okay? And so they've done all kinds of interesting experiments where they can dissociate the what from the where, right? So where something is, but you don't know what it is, or you know what something is, but you don't know where it is, and so they've figured all that out. Okay? All right, so who's taken MTF? Anyone? What, your third years, right? You take MTF in the spring? You take,
I don't know if it's class anymore.
oh, okay. ISA 2 or whatever it is? Oh, okay. All right. Anyway, so if you had taken MTF, you would learn about the eye as an optical system. So, right, in the front of the eye, there's the optics, right? There's the cornea, there's the lens, there's the iris. In the eyeball itself are the aqueous and vitreous humors, right? So all these components affect the quality of the image that gets to the retina, okay? And so if we're talking about the quality of the image, the visual image that we see, we're usually talking about the resolution of the imaging system, or when we talk about vision, we're talking about the acuity, right, of the visual system. And the acuity of vision is affected by both optical and retinal factors, okay? So there's this concept of the point spread function, right? So you've had point spread function before. Yes, good. Okay, so the idea is out there in the world, there's a point of light. That point is getting imaged on a plane by some optical system that in the simplest case consists of an aperture and maybe some kind of lens in that aperture, right? And because of aberrations in the lens and diffraction that's produced by the aperture, you're going to get a distribution of light in the image from a point of light out there in the field, okay? So that if you characterize that distribution, that's the point spread function, okay? So if we want to know what the optical quality of the eye is, we can characterize it in terms of its point spread function, okay? And so here are two point spread functions of the eye at different pupil sizes, okay? We all know the pupil opens and closes, right? And so when are you going to get the most diffraction because of an aperture or a pupil? Is this something you guys know? No? Okay. So the amount of diffraction, right, the amount of scattering because of the aperture varies with the size of the aperture, and it gets worse as the aperture gets smaller and smaller, okay? And so what happens in the visual system is that as at daylight levels, the pupil gets smaller and smaller to bring down or to adjust, compensate for the light level. And what happens is you get a point spread function due to that diffraction. But what it means is that the visual system is operating near its ideal limit, okay? So the visual system in daylight is essentially as good as it can be in terms of its image quality, its optical quality, okay? So the contrast is at nighttime, right? So at nighttime, what has to happen is the pupil has to open way up to allow a lot of light in, okay? And so when the pupil opens up, more light is coming through the lens, but the lens itself has aberrations, and so you get scattering of light because of the aberrations in the lens, okay? And so at nighttime, right, with a large pupil, the point spread function of the eye actually increases, right? But it does so, I mean, it increases somewhat, but it does that because otherwise you wouldn't have any vision at night, right? You wouldn't be able to see if you kept this small pupil, okay? All right. So anyway, the the high point is that the visual system is doing the best it can, given the wide range of conditions we have to function under, and the quality of a lens that you can make out of cells, right? So, okay, so let's stop. Oh, yeah, yes?
So when is differentials like the least, like whether the least amount of diffraction size, larger than small?
So the most amount of, so the diffraction increases as the size, as the pupil size gets smaller, okay? It decreases relatively as it gets bigger, right? So, remember like,
I'm just confused with the wording, sorry.
okay, so aberration limited for large pupils, diffraction limited for small pupils. Okay, so what this is saying is, in the visual system, the quality of your retinal image is limited by the aberrations in the lens when the pupil is large, and is limited by diffraction when the pupil is small. Okay. Okay, let's stop here and we'll start up next time.